Good morning and good afternoon to all of you for joining our live webinar today. High throughput functional evaluation of melanocortin compounds at the MC4R KIR 7.1 complex, bias signaling at the melanocortin 4 receptor. Additionally, we will have a second presentation entitled SEPA-based evaluation of pro-arrhythmic risk using the SynchroPatch 384PE. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's event. Joining us as speakers will be Ciria Hernandez, Assistant Research Scientist, University of Michigan Life Sciences Institute, and Yuri Kurashev, Principal Scientist, Charles River Laboratories. Today's presentation is as follows. Ciria will provide a look at the discovery of G protein independent pathways for MC4R signaling through KIR 7.1, opening new venues for the exploration of new signaling pathways by GPCRs. Yuri will then proceed with his presentation that looks at cardiac arrhythmias, specifically the work he and his colleagues at Charles Rivers are looking at as it relates to the evaluation of several drugs that may pose pro-arrhythmic risk, including antimalarials that were used off-shelf during the first wave of COVID-19. The evaluation and assays are run in accordance with the SEPA protocols, whereas HERG, CAV 1.2, peak and late, and NAV 1.5 late were evaluated. Lastly, we are joined by Dr. Maria Barkmas, Senior Scientist, Product Manager, Nanion Technologies, who will present a novel approach in transporter drug screening and development, specifically real-time detection of minute transmembrane currents generated by low turnover proteins, such as transporters and membrane pumps, offering new possibilities in compound screening and hit validation. Upon completion of the presentation, we will hold a live Q&A session with the presenters, and as such, welcome you to submit questions at any time throughout the course of the presentation. You can log a question in the chat window found on the right-hand side of the screen via the menu bar. Your messages will come directly to me and will be added to the queue. If you happen to experience any technical difficulties at any time, please inform us via the chat box. Additionally, today's presentation will be made available on demand shortly upon completion, inclusive of a transcribed copy of the Q&A session. I will now turn over the presentation to Ciria for the start.
To stop other screen sharing, do you want to continue? Okay. So thank you very much for attending this event. So today I'm going to talk about the bias signaling at the melanocortin 4 receptor, looking for high throughput functional evaluation of the melanocortin compounds at the melanocortin 4 receptor and the inward rectified potassium channel complex. So just at the beginning, give you an idea of this channel. So the inward rectified potassium selective channels are encoded by at least 15 KCNG gene family. They favor the influx of potassiums. So these 15 mammalian genes produce results in 7 distinct care channels that share structurally related. Being said that only the family of the number 2 and number 3 are show very strong rectification. So usually, if you look at the crystal structure of the inward rectified potassium channels, we have 2 cytoplasmatic gates bounded by the helix boundary crossing. We have the G loop, and the KIR 7.1 channel is the latest discovered. This is the least that show a very weak rectification. Also, the single conductance is very low. Lack block by external barrier incision, but the reintroduction of arginine, a very conserved arginine at the position 125, restore the strong rectification of the channel, also the blocking by external variant and cesium. So this channel, the mutated channel for the R125, is the one that we use usually to record in wholesale recording, being saying the manual or conventional patch recording on the automatic recording. So this channel, interestingly, is a high expressed in a very specialized section of the eye, the retinal pigment epitheliums, which is plays an important role in the retinal physiology. This is a channel that partner with transported to in the apical membrane of the RPE cells and maintain the potassium homeostasis around the photoreceptor out segment. Usually the function of this channel by mutations induce the degeneration of the RPE, which leads to some of the eye pathologies known as a snowflake vitreoretinal degeneration, and also the Leber's congenital amaurosis. Something that we are interested in is that the channels not only are expressed in the eye, but we can find it in other places. Especially for us, this has been described in the hypothalamic neurons. So where the NC4 receptor also is found. So usually the NC4 receptor, which is the center of the energy homeostasis in the hypothalamus, is expressed in the paraventricular nucleus of the hypothalamus. So this is the circuitry which is really important to keep the energy of the regulation of these channels. We have in this arcuate of the hypothalamus, we have two primary input components of the central melanocortin system. So the neurons that are expressing the proprio melanocortin or PMC and the HRP or agouti-related peptide. So this these two neurons release the two natural agonists-antagonists for the MC4 receptor doors in the PDH neurons. So usually when we find adipostatic signals, activates the second order of the MC4 expressing neurons in the PDH. Through the activation of the PONCI neurons, we release the alpha message to these neurons, while the HRP neurons are inhibited. This is, on the other hand, when you have a fasting condition, the firing of the HRP increase, resulting in release of HRP and GABA, and we have the blocking of the NC4 PDH signaling. Interestingly, in these important neurons, has been reported that the channel in the receptor are co-expressed distinctly in the PDH neuron, which make us think that this complex can be found or is physiologically relevant. So the melanocortin 4 receptor has been targeted for obesity because it's the most common form of found and genetically linked obesity in humans. Remains a target for the treatment of obesity because of its central role in energy homeostatic regulation. But today, most of the drugs discovered has been failure due to the unwanted target medium pressure effect in clinical trials. However, there is an exception. There is this cyclic alpha message derivative, which is called the segmelonatide, has been approved in therapy that the PONCI deficiency and also Prader-Willi syndrome. So we're going to try to look at this regulation, the complex into the receptor and the channel. So for that, just looking at the recent solver structure of the human NC4. So the NC4 is part of the rhodopsin-like class A branch of the 7 transmembrane spinning domain. Usually it's most related, even though this belongs to the GPCR family, it's more structurally diverging of the GPCRs. And we solved the structure with the SHUNA11, which is an antagonist, it's a cyclic antagonist. So the receptor is being compressed in an inactive form. So this panel to the right demonstrates what the different component of the regulation of the signal transduction through the NC4 receptor. We have G coupling receptor, the GS. We have the beta-arrestin receptor. We have also some melanocortin receptor associated protein RAP2, and the interaction with the channel we are interested. So what is this bias signaling that we're talking about? So they can be encoded through three general mechanisms. In the panel on the top, we can see that if we have in the presence of a balanced agonist that bind to a balanced receptor in an unbiased system, this may displace the regulation display two different pathways, the equivalent potency. We don't have preferences for any of these pathways, but in the presence of a biased ligand, the ligand results in different conformational responses to the receptor that can favor one pathway, single pathway, or another. We will have a biased receptor. This is in the case where you have a mutated receptor, receptor with lack of site of phosphorylation. We have also the signaling favor for another one signaling pathway, or we have a system with different components of the signal transduction is a different expression for different tissues. It was also this bias effect. So in the canonical system, the melanocortin 4 receptor is activated. You have the alpha melanocyte stimulated hormone, or alpha MSA, which is the natural agonist for the receptor. We have the agouti-related peptide, which is the natural antagonist. Usually, the activation of the receptor leads to the activation of the G protein stimulatory GS that increase the cyclic MP and also activation the PK8. But we are interested in this independent G protein pathway through the channel that's been recently reported, right? So what is some in vivo evidence that support the existence of that complex? And it's been described that in the paraventricular nucleus of the hypothalamus, or the PVH, in the second order of neuron that we talk about, the infusion of the alpha MSA, which is the natural agonist for the NC4, close the channel, which leads to depolarization of the neurons and increasing the firing, while the infusion of the HRP opens the channel, which hyperpolarize the neurons, decreasing the firing. So this is another result that I'm showing here, but another information, piece of information that corroborates the importance of the regulation of the signal pathway through the channel is when the channel is the lack of the expression of the channel in the PVH neurons. We have the lack of response of the activation of the alpha message, which means that this could be an important role in the signal pathway through the NC4 receptors, right? We have another in vitro evidence that corroborates this functional coupling of the channel and the receptor. And we, over the years, we have been accumulated a large body of work indicating that there is a direct functional interaction between the NC4 receptor and the channels. So we solved the crystal structure of the NC4, and while assessing the different construct of the receptor, the NC4 receptor, we use a construct that have an enzyme in the intercellular loop of the receptor, which block the coupling with GS. While block the coupling to GS, we found that still this construct is still coupling the function of the channel, which means that it's independent of GS, it's totally direct to the channel. Another piece of information with this is we use a mutant that block the binding to the alpha message or HRP, and also that mutation blocks the function of the channel. So these findings confirm that the NC4-dependent coupling of the channel with the construct used in the crystallization. So this diagram displays this independent G protein interaction between the channel and the receptor. So how we would explore this NC4 receptor here's endpoint bias signaling pathway? We have to demonstrate different response in different assays. So until now, we have no highly sensitive real-time high-throughput screening that has been documented to identify NC4 agonist, antagonist targeting this complex. So in this study, we're proposing, we're using the automatic patch clamp recording platform through the synchro patch to evaluate the coupling of the NC4 receptor and the channel based on the real-time detection of the channel responses. And we use another assay, which is a split luciferase cyclic reporter or the cyclic glow sensor to demonstrate the bias response. So we propose to identify compounds that functionally targeting the coupling between the complex, trying to identify novel therapeutic alternative for the treatment of obesity, So we have these two assays to fully characterize and validate compounds that functionally targeting the coupling between the channel and the receptor. We have these two settings. We have, to the left, we have this akinetic plate reader through the Hamamatsu Phonoris. This is the FDSS 3000 that allows a real-time simultaneous monitoring of the luminescence in the 384 well plate formats, similar to the one we have for the synchro patch. So, so we use, we adapted a commercially available split luciferase cyclic impede reported assay, and we measure the cyclic impede responses mediated by the NC4. We have a hexel line that express the glow sensor, and we measure the kinetic profiles of the responses in the EC50s concentration response curve of mesate in the presence of the HRP, right? And to the left, we have the synchro patch settings, where we have this platform that's capable to measure 384 whole cell recordings, at the same time, and use to evaluate the bias signaling through the complex. So for the recordings for these studies, we just compare what we use for the manual patch clamp, the conventional patch clamp, and the synchro patch. So usually when you use whole cell recording in the conventional patch clamp, we use hex cells that are transient transfed with the construct, the channels of the receptor, right? We record 48 hours after the transfection. For the synchro patch recording, we use a stable cell line that flip T-Rex hexagon lines that express either both the receptor and the channel of the channel. We are using the channels that are mutated, that display large current, and they're able to block by barium incision. This system is selected with agromycin, and we record the channels after 24 hours induction by doxycycline. So to give you an idea of comparing the result we have in this panel to the left, the representative currents holding cells minus 60 millivolts go to minus 180 millivolts to 0 millivolts. It's a very huge rectification we have because the internal solution contain magnesium. You have this inactivation, very fast inactivation, which is telling us that the channels recording is with the strong rectification. The voltage reverse potential is about 66 millivolts. Comparing with the cells, the T-Rex cells expressing only the channels or the channels together to the receptor is very similar. The traces that we obtain they have this inactivation by magnesium by the internal, and we have a very nice rectification similar to the manual the way obtained in the manual patch clamp recording, and also similar voltage and reversal potential for all of them. So for the initial studies, we set up a two-stage voltage protocol consisting in after holding a minus 60 millivolt, we have a step hyperpolarization step to minus 140 for 200 milliseconds. And after we have this ramp, and usually we measure the peak current amplitude during and after compound application, and that step minus 140. Just looking here in the panel to the lower, we have no effects during the reference solution, and these channels are externally blocked by external barium. Also, another characterization is the channels, they are not blocked by TA, which is a blocker, voltage-gated potassium channels, not the rectifier. We have a strong block by cesium, external cesium, and two other more specific inhibitors for the KIR 7.1 also show very nice block by these compounds. So initially, we tried to find what is the responses if we find biased response to different mutants of the HRP. The HRP is this classical antagonist, and looking at the structure of the HRP, it's been described that the outer part of the HRP, which are in blue and red here, can modulate the allosteric conformation of the receptor and may be biased to the responses to the channel. So we find out that we have, comparing with the wild type, in 3 different mutations of this segment of the HRP protein. You look into the glow sensor assay, the CRC for the cyclic P is very similar for all of them. But if you look into the responses for the channel, which is expected because the HRP opens the channel, assessing the opening of the channel, comparing with the wild type, we found that the mutation, the replacing for 4 lysines to the outer segment of the HRP peptide usually increase the affinity, the response towards the channel, which confirm the bias signature to the HRP through the channels. So also make a characterization of the classical activity of the peptide. Some of the melanocortin ligands, we have the HRP, which is a natural antagonist for the MC4 receptor. We have SHU-911, which is an antagonist, which is this cycled lactam antagonist, which is made by this core, which is the alpha message. Alpha message is the natural agonist for the MC4. It's just the introduction of D-naline in this position transform agonist to antagonist. So in the assessing the second response, we have the expected decreasing of the response with HRP and SHU-911, and the alpha message is increasing the levels of cyclic MP. But assessing the answers, the responses in the synchro patch with the channel, we found that HRP is a bias because while it's decreasing the levels of cyclic MP, we have an opening of the channel. SHU-911 also similar response as HRP, showing that this is also a bias compound or drug to the channel. And alpha message is the classical agonist that close the channel and also increases the response to the receptor. We evaluate another non-small molecules to try to identify if this is the case. We have to can find another bias compounds that regulates this complex. We use these three melanocortin antagonists that all prevent that being studied and prevents the loss of lean mass in different models, and also induce weight loss in mice, all of them. So in the glow sensor assay, all the antagonists decrease the function, the levels of the receptor. But we evaluate the responses to the channel through the synchro patch. We find out that only this molecule act as a bias, but while decrease the cyclic MP responses, opens the channel, similar to that we found to as HRP and SHU-911. The others two are usually or similar to the responses as alpha message. So surely we found that we can't find different, we have been trying to find different, a series of melanocortin receptor peptides that behave like a second melanotype, which is agonist, HRP, or SHU. It was being, well, I'm not gonna show all the results, but we have some nice compound that show very nice bias answer. So in brief, just in conclusion of this talk, we have the ambivalent nature of the HRP different peptides can be easily demonstrate this G protein independent coupling assays, right? Being biased towards the channel, not to the GS coupling. SHU-911, which is a very classical antagonist, this is they contain this D-null in the cycle, maintains the same profile of the HRP, which is was a surprise. Is doesn't signaling through the GS coupling to the receptor. So also we found for assessing the different peptides modification of the core sequence and the message agonist and somehow make different responses to the channel opening. It favors some receptor conformation that favors some of the signaling towards the bias to the channel. Also, this variation can be mimicked by some of the NC4 small molecule that's as proved at the NCT24. And it's interesting that we can demonstrate this effect through these two small molecules, and this is an idea that appears to be associated with the GS coupling agonist. We're not sure about that, but probably we can see if this coupling impacts depressor activity if we do model studies in animals, right? So another step that we can looking at, we try to validate our assay. So we next we are using small molecular compounds for the draw reproportion set to the university to try to find novel compounds that can easily screen and validate the very fine bias signaling through the channel. So that thanks to finally to all the collaborators and friends that involved in these studies. Thank you very much.
Thank you so much, Ciria, for going over your presentation with us. I now have the pleasure to introduce Yuri Kurisev from Charles Rivers.
Jason, can you confirm that you can hear me?
Yes, we can hear you loud and clear, and I can see your
Okay,
slides.
thank you very much. Sorry for this little bit absence of feedback about my sounds. My presentation a little bit more. I am from Ciria, from Cleveland location of Charles River. It's formerly Chantest, and our whole history was to study different pharmacological effect of very variable compounds on all possible channels that we can generate cell lines. But still, for many, many years, HERC and cardiac safety in general, it's our major target. We do this a lot of studies related to cardiac safety. We have a long history with automated patch clamp. We started with Patch Express, and then we had a Quattro Ironworks, and then Q-Patch, Ironworks Barracuda, and finally we got 2 years ago SynchroPatch. Now it's our one of major platforms in parallel with Q-Patch. Also, Charles River has a Cube, but it is not in Cleveland. Okay. Now we have many assays for different cells, but different channels. But still, as I mentioned, our favorite for cardiac safety is HERC, calcium 1.2, cardiac calcium channel, and sodium cardiac channel now 1.5. And we're doing this different assays that could be customized for customers. And SynchroPatch is our most adjustable platform. For manual patch clamp and for Q-Patch, we have more standard approach, but for SynchroPatch is still in development, many things, and we're very flexible. And this instrument is a little bit advertising. I hope it's okay with non-19. Very convenient for many approaches. We can do end safety studies and discovery studies, and we can fit to many customers' interests and things. And we using physiological temperature now at a regular basis, and also 384 plate-based experiments give us a very big advantage for control reproducibility. We have a positive and negative controls in every plate that give us options easy to compare studies from a year ago and 3 months ago and last day. Very efficient keep to track of projects. And of course, it's we're doing HTA-like projects when we screen big number of compounds and single concentrations, but also we very often we have studies with those responses for selected compounds. HERC is a basis for cardiac safety. It's for many years we work with it, and I will use HERC to illustrate some possibilities what we have can get, and we can propose to for as part of our service. HERC has a long history, and this is two to test pulse voltage protocol is really has 20 years history when you depolarize cells to relatively positive potentials, and then you're measuring tail current, and if you see drug effects on effect of tail currents. And for a long time, room temperature, this protocol was used at manual patch clamp and Q-patch and patch express and everywhere. Now, in automated patch clamp with synchro patch, we start to use physiological temperature. And we found that it's not difficult. It's no anything. It's not changed that like success rate or other things. We can use it on a regular basis without any problems. But also, this is an example of protocols that SIPA recommended and set it. And this is, for me, is an example that we can adjust protocol for customer needs. If somebody recommended this protocol, we can use it without any some validation, characterization, and then it's in our library of voltage protocols. And here, this is continuation. In previous slide, we see that we have no problem with vehicle control. It's stable recording for 5–10 minutes. This slide illustrates that how temperature can affect drug effects. At room temperature, the phthalate have very slow onset of inhibition, and at the physiological temperature, it's give much faster. It looks like a temperature potentiate effect of the phthalate if you measure it at fixed time. If it stay 5 minutes, temperature effect of the phthalate will be much potent. In manual patch clamp, traditionally, you have a unlimited recording time until you have some kind of steady state. In automated patch clamp, especially in DAT4 format, you should set limit for recording because then data analysis becomes very uncertain. If you take one cell after 3 minutes, another cell after 10 minutes, it's okay for very specific studies, but for really high throughput not makes sense. And in this case, we can say that temperature really could be very critical. For some compounds, you can get much more accurate data because this kind of process going faster, inhibition faster at physiological temperature. And as another addition that I can mention that 2-pulse protocol and SIPA pulse run protocol gives the same. In numbers we show later, and we see in the time course of onset of inhibition will be about the same, and only we can say that all previous data collected with 2-pulse protocol not using a SIPA protocol not modify them too much. And here I make special showing that we have 2 protocols, 2-pulse protocol, that we're using for many years on all platforms, such as manual patch clamp and Q-patch standard protocol, and then this new SIPA protocol that also could be used on all platforms. And this historical data from inhibition with cisapride, with step-run protocol, SIPA protocol, and classical 2-pulse protocol. And no difference. It's everything. It is some variability, but really very condensed, and it's each point. It's indicate independent experiment. C-Sepride was used as positive control in some cases it was special validation experiments, but all this coming from 8 concentrations dose response curve with 4 replicas per concentration. In some cases we lost some cells, but usually it's not less than 3, 4 replicas per concentrations, and you can have idea about how this data looks like, and dispersion through few months. Also, it is historically we have CHO cell lines, HERC expressing CHO cell lines, and HERC expressing HEK cells. It is different reasons, and sometimes it's depend of a specific study which cell line we used. Historically, CHO was preferable for patch express, especially for Ironworks Quattro. Ironworks Quattro was very difficult with HEK cells, at least in our hands, but CHO was okay. And Barracuda, Ironworks Barracuda was also prefer CHO cells. For Syncropatch, we don't see really technical big difference. Both cell lines works well enough. At least again in our side, we have no problem to switch between CHO and HERC, CHO and HEK depend on a very specific study. Like for transient transfection, HEK cells much more efficient. For stable cell lines, it's CHO could be preferable because they have less collection of endogenous currents. Also, for HERC, it's not important, but for sometimes when you handle calcium and sodium channels, endogenous currents can give you contribution. And right panel showing that we have different conditions like CHO room temperature, CHO physiological temperature, HEK room and chain physiological, and this no significant difference between pairs, at least when we compare. Could be some difference could come from how drug-resistant transporter works in these cells. It could be temperature effect sometimes for more efficient pumping out of drugs from cells, and in HEK cells it's maybe a little bit more efficient compared to CHO cells. But in general, when we select between CHO and HEK, it's for HERC, it's we can firm say that it's no difference, but for other channels, it will depend of this specific cell background, and especially, of course, in case of transient transfection, HEK much more convenient. This is one more indication how it works in case of temperature. With historically, it is a two well-known published drugs that have temperature dependence. Erythromycin, we can say yes. We can say that in the physiological temperature effect of erythromycin erythromycin more potent at physiological temperature compared to room temperature. For Sotalo, it's not so clear. It is some trend, but it's not significant. And explanation could be sometimes you have an issue with solubility, and the physiological temperature improves solubility, and this can improve the inhibition. And plus, again, it could be issue with pumping out of drugs. in some cases, it's physiological temperature. It could be more efficient, and not the fact that physiological temperature give you improvement in any case in the sensitivity. It could be a little bit opposite. I'm not illustrating this case, but some drugs we have this trend that physiological temperature is potent than room temperature. Okay, I demonstrated this is what HERG in our site can do, what we can do with HERG. And this is a little bit summary historical that we compare our synchro patch at different temperature with Ironworks and Q-patch and manual. Q-patch and manual is published data, but it's from our site. Some people like did all this experimental across whole panel. And without the specific details, we can say that with HERG very close results between synchro patch room temperature and Ironworks Barracuda room temperature and manual room temperature. Historically, we can say that if we switch from this whole project will demonstrate that if we will switch from one platform to another, it will have no huge difference. Yes, synchro patch give us advantage with temperature. It's more flexible with protocol, continuous reporting if we were talking about Ironworks Barracuda. But still, our data collected on other platforms comparable with synchro patch, and it's very important for many customers that have a long history with us. And this is to say that we're using everything that synchro patch provides as new, especially internal perfusion. This is the best way to use this platform, but still, it's not annihilate our previous results. And as illustration, how this for cardiac safety application of this everything safety. We participated in project about several anti-malaria drugs, how they cardiac safety approach. It's this paper. It's now available online, and what I will show it is not shown in the paper because it's rarely in paper. We have only numbers. This is only what this part of table is published. All other it was a supplementary data that not easy to see. And we tested in this project two anti-malarial drugs, chloroquine and hydroxychloroquine, and couple antibiotics that are regular base used sometimes together with other drugs. The idea was to see how these two anti-malarial drugs, it was known that they prolong QT and could be polyrhythmic, to see what is the ion channel effect pattern for these two compounds and in combination with antibiotics. And everything was done as much possible as CIPA recommended. We use CIPA protocols. We use temperature when possible. And was tested combinations of azithromycin and chloroquine and other things. And it is also important when at least it's helpful to remind that each experiment for these drugs had a positive control, and we have the positive controls for every experiment. For calcium channels, we did 2 experiments, and data very close for positive controls. For drugs, it's combined. And as we saw before, that variability between experiments low, and data from one experiment makes sense. And we have positive control to confirm that experiment was normal. Plus, it's also useful to remember that besides positive control, we have vehicle control in the experiment, and this gives us an option to make a correction for rundown. Because rundown or sometimes run up variable, and one of it's complicated things to make a rundown correction. And 384 plate-based experiments very convenient about rundown. The results adjustable easily. This is illustrations. It's not published, but it's data. In previous table, this data. It's our favorite. Okay, it's our SIPA way, SIPA protocol. What we see with it? It's what before. It's our reservoir vehicle. It's chlorquinine and hydroxychloroquine, about the same. You can see that at onset of chloroquine is faster compared to hydroxychloroquine. And those responses, it's 1 micromole for one compound and about 2.5, 3 micromole for another compound. And based on clinical trials, you can say that chloroquine is more toxic than hydroxychloroquine. And also this it's a dangerous from point of view estimations because it's maximum free concentration in blood peak could be about 0.5 micromole. It's very close. It's not a surprise from her data that this drug very proarrhythmic. Sodium channels. We use this assay on a regular basis, also it's part of safety. But peak current usually not too many drugs that affect peak current. It's a block, but as you can see that IC50 values the same, chloroquine and hydroxychloroquine much. This drug much less potent for sodium peak current. But we have no any problem doing this recording from Ciria patch. It's also our favorite channel. Late sodium current. This is a much more important component of proarrhythmia safety because it was demonstrated already many times that the inhibition of late sodium current could be make a drug much more safe if it's despite of the effect on Kirk. And in this case, effect not very different from this is peak current. It's about 14, 17. Late current about in the same range, but it is a clear inhibition. And from point of view of our capacity to measure late sodium currents, it's very efficient. We activate late sodium currents in CTX, and then co-application of any drug give us effect, and then high dose of inhibitor, lidocaine, simply give us zero potential. And the SIPA protocol, it is, as we saw before, it has phase of voltage step for the current, peak current, and the ramp decrease for measuring of late sodium current. At the temperature, works very well. calcium currents. It's most complicated. I think many, yeah, it's well-known thing that with calcium currents not so easy. First of all, it says, of course, the rundown cells and buffers, what if works well with the calcium currents. But sometimes rundown too fast. After 5 minutes, the current is difficult to measure, especially when it's about late sodium, late calcium current. But these two drugs, it's chloroquine and hydroxychloroquine, demonstrated very interesting. No, it's not something very new. It's known before, but the fact that this is a drug produces a really tonic block. When you activate current from holding potential, the drug inhibits it very well. It's like sodium, about 20 micromole. But then it's what my vision that it's a LiPo block, and instead of deactivation and activation, currents coming back, and really we have increase of late calcium current. That I saw a couple of other drugs that works the same way, but it's not typical way. Usually, like for example, nifedipine, you have no difference. It's proportionally block both end peak and the late current. But these guys, chloroquine give a apparent potentiation of calcium currents. I think it's not really potentiation. I think it's simply a LiPo block, and it's a delay inactivation and calcium-dependent inactivation. And the same we found for related hydroxychloroquine. Also very nice. It inhibits peak current, but it can potentiate late current. And in theory, this should make this drug very toxic from cardiac, very pro-arrhythmic, but still people who use it as anti-malaria for anti-malaria treatment, people are alive. Therefore, from point of cardiac safety, this is very questionable. That in coming back, that maybe late sodium current contribution could be very critical too. That if you block late sodium, maybe remove effect of on late calcium. I'm not sure how this whole modeling reflects this well, but it is a couple abnormal drugs because classical positive controls, what I saw, they very straightforward block end, peak, and late currents. And conclusion based on electrophysiological data, we can say that we can separate the drug, one drug more potent than the other, and this is, for this particular study was exactly very close confirmed with another group, with Q-patch done in another lab. If you have specific interest about numbers, you can find in the publication. And this is atypical drugs. Maybe at some point I will do more detailed analysis of calcium current, but it's interesting example what could be when you're looking for many drugs for cardiac safety. This is how we have really all this particular project done with helps of Kayun Bo, myself, and Jim Kramer, our manager and head of automated electrophysiology, but just in case for the advertising, I can remind that we have also ligand-gated team with Nikolai Fedorov as the head, and we have many other channels and plus flippers and everything else. I think it's end of my presentation, especially I see that it's noon exactly. And if any questions, I can respond anytime.
Thank you so much, Yuri, for going through your presentation. I will then now ask Maria to prepare her slides, who will look into a novel approach in transporter drug screening and development. So I hand over to Maria.
All right. Thank you, Jason. So I would like to close this webinar with a very short 10-minute outlook on a novel approach in drug development for electrogenic transporters. So as you are all electrophysiologists, you are for sure aware that ion channels are not the only protein generating membrane currents. So secondary active transporters and pumps also generate bioelectric currents. So pumps consume ATP to move ions, while secondary active transporters use the resulting ion gradient to enable uphill transport. There are already more transporters described only in the SLC family than ion channels, and they become increasingly interesting for pharmacology. The problem is that the turnover of these proteins is usually many magnitudes smaller compared to ion channels. So Charlie here demonstrates that for us. So the very simplified explanation is that ion channels basically open the gate, while transporters change the conformation in each transport cycle and leading to a very low transport rate. Anyhow, they generate currents, so it would be nice to use electrophysiological methods. So if they, as they have many advantages. I don't need to go into detail before this audience. So instead, in particular in drug development, other methods like fluorescent or radioactive uptake assays are usually used because the currents are just too small to resolve with an acceptable signal-to-noise ratio. A way to compensate for that is, instead of just measuring one cell or a handful of cells, to use a sensor coated with the membrane of thousands of cells. And this can be done with a method called solid-supported membrane electrophysiology, or short SSM-based electrophysiology. It's called like that because the SSM is linking membrane fragments or liposomes to a capacitive cold sensor. I would like to illustrate the amplification one can reach with this method with a little experiment we did a few months ago. So what we did is we used Pepti-1, one of the transporters, one of the few transporters that you can actually measure with patch clamp, as it has an extremely high transport rate. It has a very high transport rate because its basic or major function is the uptake of dietary peptides in the intestine. So we tried measuring Pepti-1 currents with a 4X patch clamp chip, and we were actually very positively surprised by the outcome. So you can see here, there is a little current. But when we used exactly the same cells on an SSM sensor, we get this very nice 10 nanoamp currents. So if we set that into scale, we see that with SSM-based electrophysiology, we can actually reach an amplification that enables us to measure transport currents. Of course, this method has different pros and cons compared to patch clamp technology. For example, it's not possible to establish voltage clamp settings. But for transporters, this is not necessary, as they can be activated similar to ligand-gated channels by applying the substrate with a perfusion. The experimental scope is actually very similar to patch clamp. It's possible to analyze the current shape and kinetics, as well as the influence of environmental conditions. But for pharmacological approaches, it is, of course, most interesting to investigate substrates and inhibitors. And the nice advantage of the sensor-based technology is that one can record from that one has not to record from viable cells, but from isolated membranes. And that means you can prepare them in batches, freeze them, and it does not make any difference if you record from plasma membrane, internal membranes, or even bacterial membranes. So SSM-based EFIS is actually not very new. It's been around for some time now. However, so far it was never widely used in pharmacological research and drug development because the throughput of the method was very, very limited. So with the Surfer 96 SE, we have the first full parallel automated system operating on a 96-well format. It has a very high success rate and reproducibility, and also the preparation of the sensor and the workflow are all automated. So that means a throughput of several thousands, up to 10,000 data points per day can be reached easily. And therefore it's qualified for some applications in drug development. For example, it can be used in hit validation. So I would like to illustrate this with a case story from the Department of Integrated Drug Discovery at Sanofi, Germany, with friendly permission, of course. They performed an inhibitor screening on a sodium-coupled amino acid transporter using a membrane potential assay on the flipper. And they developed a secondary surfer assay to validate the hits that came out of that screening. So the goal was to have a direct assay with high quality data to characterize the compound effects and most importantly to filter for false positive hits. So here you see some control experiments. They used a CHO cell line expressing the transporter. They prepared crude membranes and then activated by applying the substrate. So when either sodium was not present or the amino acid was not present, there were no signal, and also in the wild type, they did not see any response. The success rate was very high, so I don't have the quantification here, but you can see an example of an experiment where you see very nice homogeneous currents over the whole plate. So what they did first is they looked at different amino acids to identify substrates of the transporter, and they compared also the results with different more conventional transport assays like a flipper, radioactive uptake, and also to electrode voltage clamp in oocytes. And you can see the results are quite comparable. So finally, they chose one of the substrates for the inhibition assay and used a well-described tool compound to verify the inhibition assay. And finally, they used a whole set of compounds and compared the results with a mass spec assay to prove the functionality of the SSM assay. So after this validation was successfully completed, the assay was actually included in the workflow, and the Surfer was indeed a valuable tool for them in characterizing the hits of their screening assay. Of course, I cannot share this data with you, but the project was completed, and now the whole story is about to be published. The manuscript is accepted and should soon be out if you're interested to learn more about that. With this, I will finish my short introduction, and if you're interested to learn more about this technology, you're very welcome to join our webinar next week, where 2 of our users will present their scientific applications using the Surfer N1. This is the little brother of the Surfer SE. And as an outlook in September, we are also hosting a webinar with pharmacological focus and SSM electrophysiology. So thank you for listening, and I hope this was an enjoyable outlook for you in the field of membrane transporters. And with that, I will hand over to Jason, who will moderate the joint Q&A session for all talks.
Thank you so much, Maria. I will go ahead now and moderate some of the questions that we've received. So yeah, initially during Ciria's presentation, there was a question that came in regarding success rates for your experimental runs. Did you look at the reproducibility for the different assays?
Oh, yes, yes. So you mean about the percent of the success of the recordings among the cells, right? So, yeah, so it was between 70 in 75, 90%, depending of the day, depending of the health of the cells. Yeah, but it's pretty nice reproducer between the cell lines that we were using. Yeah.
And then another question that came in regarding sweep intervals. So which sweep interval did you use in your CAV 1.2 voltage protocol? Or that this was for Yuri. That was a question for Yuri. So sorry about that.
Sorry.
Let me see if Yuri is on mute by any chance.
Okay, now you hear me?
Yes, we can hear you, Yuri.
Yeah, for CAV 1.2, usually we're using 10-second interval, but sometimes we are also using 5-second interval. This is most cases work about 10-second standard case.
And then also regarding your success rates for your experimental runs and the reproducibility for your different assays.
Success rate very depend of cell lines and the channels with HERG, for example, and other potassium channels, as well as in sodium channels, like 1.5. Success rate close to 90%. In best cases, we have 95%. But normally, we considering that experiment successful, we have like cut off 80%. If below 80, we consider that the experiment failed. For calcium channels, this is a little bit different. First of all, it's dependent of cells, let's say cell quality. In some cases, we have a success rate again the same 80%, but in many other cases, like with SIPA protocol, to measure late calcium current, we need to really increase the number of replications at least to 8 instead of 4. And this sometimes with calcium channels, physiological temperature and SIPA protocols, that there's 25 success rate, it's not bad. Because if we work against run down, and run down in many cases. But in good days, success rates well above 50% for calcium channels. For HERG, again, I say, and this is for sodium, it's 80% is minimal, and then 90% is standard. Reproducibility, as I demonstrated in this HERG validation slides, that usually the difference between IC50s for HERG and sodium channels less than 3 times. It's minimal significant ratio, range between 2 and 3. It's well reproducible. Some cases it's dependent of protocol, and when strong voltage dependence in the subject of study, variability could be high, but normally below minimal significant ratio, below or around 3. If we have good quality, everything good quality, reproducibility very good between, you can get the same result with 1 year interval without any problem. It's, we have many customers that include some reference compounds in studies, and this could be open label that it is a reference compound. It could be more as any compound in different name. And usually we have no the problem with reproducibility as we considering it for plate-based assays. That 8 concentrations for replicas usually give us very close numbers with 1 day interval and 1 month interval and 1 year interval. We have already collection for syncropatch. And positive controls also we keep track, and our goals to have a minimal significant ratio about not more than 4, but in major cases between 2 and 3. Something like this.
Okay, perfect. And then I'm aware that we're running a little bit over time, so unfortunately some of these questions that we received would have to go through in the transcribed version. So one closing question for Ciria. Regarding your experiments that showed direct coupling of the MC4R to KIR 7.1, what are the physiological implications of your assay in terms of feeding behaviors and energy expenditure?
Well, we still don't fully understand the implication for the direct MC4 receptor KIR 7.1 coupling in a live organism. What we do know is that both are present or are expressed where they need to be, which is the MC4 neurons of the paraventricular nucleus of the hypothalamus, right? I personally think that the direct coupling could represent an escape mechanism to control, to some extent, the function, excitability of these neurons under certain circumstances where normal cyclic MP-induced responses are shunted. For example, in PONC deficiencies, right? So the fact that these are 2 independent modular functions is a testament of this, and one can envision manipulating one pathway pharmacologically without affecting the other by biased drugs.
Perfect, thank you. So again, for my part, one last big thank you to Ciria, Yuri, and Maria for their presentations today. To everyone that's still in attendance, you will shortly receive an on-demand copy and a transcription with the full Q&A as well as the full session. And we thank you for your attendance and wish you a good rest of your day.